DNA is the master blueprint that directs how every cell in the body is built, maintained, and replicated. Since the discovery of the double helix structure in 1953, we've understood that biological development follows an encoded architectural plan rather than spontaneous assembly. Because this code guides each generation of dividing cells, maintaining DNA integrity is essential. Even small errors in the blueprint can compromise cellular function and, over time, human health. Genetic toxicology serves as the safeguard for genomic integrity in pharmaceutical development and chemical safety assessment. Its purpose is to evaluate how physical and chemical agents interact with DNA, identify threats to the fidelity of the genetic code, and act as an early defense against potential cancer risks or heritable genetic defects. By integrating molecular biology with regulatory science, the field ensures that new therapeutics do not introduce unacceptable genetic hazards. Genotoxicity describes a substance's ability to damage genetic material, ranging from strand breaks to chemical adducts and base modifications. It reflects the hazard, the capacity to cause harm, not the final biological outcome. Cells are equipped with sophisticated repair systems that can correct many lesions before replication. Only when repair fails and the damage becomes fixed as a mutation during cell division does genotoxicity translate into a lasting biological effect. Carcinogenicity refers to the ability of an agent to induce tumor formation, and it can arise through different mechanisms. Genotoxic carcinogens initiate cancer by causing permanent DNA mutations and are typically modeled with a linear, no-threshold dose response. Non-genotoxic carcinogens act indirectly through receptor signaling, hormonal disruption, or chronic tissue injury without damaging DNA. These mechanisms generally involve threshold effects, where risk appears only above certain exposure levels. Understanding this distinction is central to regulatory risk assessment. Genetic toxicology relies on a tiered testing strategy aligned with OECD test guidelines and ICH S2R1. Assessment begins with the Ames test, which screens for point mutations in engineered bacterial strains. An in vitro mammalian assay, such as the chromosome aberration test or micronucleus test, then evaluates structural or numerical chromosomal damage. When in vitro results are positive or specific concerns arise, in vivo studies provide physiological confirmation. This framework supports the 3 Rs by emphasizing efficient in vitro screening before progressing to animal studies. Clear distinctions among genotoxicity, mutagenicity, and carcinogenicity are essential for interpreting risk. Genotoxicity is the potential to damage DNA. Mutagenicity is the permanent, heritable change that results when damage escapes repair. Carcinogenicity is the disease outcome, tumor formation, that may or may not stem from genetic damage. By separating transient lesions from fixed mutations and clinical consequences, genetic toxicology ensures that promising therapeutics are not discarded due to reversible effects, while genuinely hazardous compounds receive appropriate regulatory scrutiny.